Ja, dat komt dan wel.
Oké.
Ja, en dan als het klaar is, stop
Ja.
record.
Okay, so good morning. I see that a lot of people are still in bed, I guess. But anyway, you are here, and I hope that I can give you some refreshment of something which I hope that you already have had in secondary school. Who of you did get some information on metabolism during secondary school? Some? Okay. So there may be a lot of repeat of what I'm going to tell you, but hopefully it will help you in approaching the next case that you will start today, which is about metabolism. So far in the block you have been dealing with digestion and the uptake of the different metabolites once they are broken down to the final units, when they are taken up by the enterocyte and coming into the blood. That's what you see here. So I'm not going into that anymore. That's what you have done. Let's put we have to go like this, of course. That's better. So this will be the case which you will start today, or did you already start this last week? No, this will be for the afternoon. So basically what your mission will be for this week to look at what happens on the levels of the different tissues with those components that are now brought in by the blood to the different tissues. And I don't know how you will approach this in the tutorial group, but one suggestion could be that you split up in 3 groups, and one group is dealing with glucose, the other group will be dealing with fat, and another group will be dealing with amino acids, and you brainstorm about that, and then you bring back all your information that you gathered in the small groups and discuss that plenary, and then inventorize what are the different things that you really need to know more than you will know at present. Then on Thursday you will report that back, so you will discuss with each other what you all have found, and then later on, and that's not in this week, unfortunately, it's only next week, which the scheme, the timetable didn't allow us to do that this week, so I think it will be next week Wednesday. You will see me back again, and then we will discuss what came out of that tutorial group, and I will address all the questions that you still have. For the moment I will give you then the background to do this case for the afternoon. Now before I continue, I want to say that if I'm going too fast or too slow, please give me some feedback. I can speed up or slow down, and just interrupt me when something is not clear. So just ask the questions, don't wait till I have finished my whole story, because then we don't know anymore what it was about. Okay? This is what I will basically do with you. I will first start giving you some information about general principles of metabolism, and then obviously we will go and look at the different pathways which will be divided into the degradation pathways and the pathways to synthesize new components. And in the second part then I will answer all the questions that you have covered during the tutorial group this afternoon and during your study this week, and we will do a little bit more about integration of the different metabolic pathways in the different organs, and if time is left I can even give you some examples of metabolic disorders, because that's actually the moment that it becomes, well, let's say more or less fun to deal with this content. I know, I realize it's, well, not too exciting material, metabolism, but I do stress it's very important to know about this. The sources I have used was just a limited number. Devlin, Textbook of Biochemistry, which is a very good book I can recommend you. There's another book which is Actually, formerly it was called Stryer. Stryer unfortunately died, and it's now taken over by other people, but his name is still there. Another important biochemistry book, Nelson and Cox and Leninger. And then finally the book that you have been or are still using, I think, during this course, Smith and Morton, The Digestive System. And for the second part, some of the schemes I will be using in the second part are coming directly from Smith and Morton. So, we will start with the general principles about metabolism. Now, of course, we all know the terms of catabolism and anabolism. Catabolism is degradation of energy-rich nutrients, like the carbohydrates, fat, and proteins, and that will be degraded in order to extract energy from those compounds, and you're left with waste products, carbon dioxide, water, and ammonia. Now, of course, the energy that has been extracted from these components, you need to gather or to collect the energy somehow, and the energy is collected into a small set of molecules, which is ADP, NAD+, NADP+, and FAD, and they are turned into these components, ATP, NADH, NADPH, and FADH2. Well, for the counterpart of this, we have, of course, the anabolism, where you have to produce and synthesize macromolecules that are essential for life, proteins, polysaccharides, lipids, and nucleic acids. And for that you need precursors, building blocks, which are also coming from this pathway, of course, from this catabolism. And those building blocks are then brought together and synthesized into big molecules depicted here. And it will be clear, of course, that that requires energy. So that energy can be extracted again then from these components, and they are regenerated into these components so they can be used again for the catabolism. Now that's fine, but that's not all there is, because the chemical energy somehow has also to be converted into mechanical energy in order to produce labor. And that's the whole scheme. Now, this is the whole scheme of all metabolic reactions, and it will be clear that it's no way that we are going to deal with all these reactions today and even later on for you. That's not necessary. Of course, you can, if you look carefully, you can recognize some of the pathways that you do know. For instance, what would be this pathway? What's the most common pathway that we all know from metabolism, from secondary school? When you start studying metabolism, you always start with? No? Glycolysis, yeah. Photosynthesis, we are not doing photosynthesis, yeah? We are human beings. We are not plants. So, that's the glycolysis. So that would be the Krebs cycle or the citric acid cycle, and then obviously what's coming next? After the Krebs cycle? Yeah, the oxidative phosphorylation or the respiratory chain indicated here. But you see there's still a lot of pathways left, and as I said, we are not going to deal with all these pathways. This is what is the simplified scheme, which is still quite complicated, but this is what I'm going to do with you today. In small steps, we will build up this whole scheme from nothing to what you see here. And that's in fact what I recommend you to use and to know more or less how to use it when you encounter a metabolic problem. Like what we all know, what's happening to a diabetic patient. There's a metabolic problem. And you can use this as a kind of a map, just to look at, suppose you have some kind of an enzyme defect which will block one of the pathways, say some pathway in the citric acid cycle, yeah, if that would exist, then you can imagine that some of the other routes that lead to the citric acid cycle will build up, that you will get a traffic jam, so to speak. Yeah? And that's the way you need to use this map. Okay. Um, Indeed, it looks complicated, and you may ask the question, why is it so complicated? Why would it be so complicated, the whole metabolic system? Well, that's a bit difficult question, maybe. If you oxidize glucose in one step, what you can do, for instance, in a calorimeter, you know what it is? A small compartment, you put glucose in it under the proper conditions with oxygen, ignition, and then the glucose is immediately in one step burned up into carbon dioxide and water, liberating lots of energy. Actually, if you consider the energy that you require in the different reactions in the body, much less is needed than what is present in what you liberate on energy by combusting glucose. So what the metabolic system actually is doing is converting big money into small money that you can use to pay different reactions. Yeah, I guess you understand what I mean with this example. And the small currency, in fact, that is what we discussed just a few slides ago. That's the ATP and the NADH. So those are the little packages. Those is the small money to pay for the different reactions that you want to do. Then another principle of the metabolism is that it's a converging system, meaning that it's in fact an economic system. Suppose all the different components that enter your body by the food, the different components that are present in the food, like the carbohydrates and the fat and the proteins, different carbohydrates, different fats, different proteins, that for each component you would require a whole set of enzymes. That is not very economic. That would result in an enormous amount of enzymes that would be required for all the different components in the food. So the whole metabolism is organized in such a way that everything is brought down to a limited set, a limited number of molecules that can further be degraded into the citric acid cycle in order to generate ATP. So the number of enzymes that you require, of course you require a lot of enzymes here, but then it becomes less and less until with a few, still quite a number of enzymes, you can deal with the final reactions. Okay, as we already have seen, ATP plays an important role in metabolism. And what is ATP? You don't have to know the whole structural formula of ATP, but you should know that it is composed of an adenine unit, a ribose, a sugar unit, and 3 phosphates that are attached to each other, and that's where the energy is contained in the molecule of ATP, because upon hydrolysis of each phosphate group you liberate a certain amount of energy, and you generate then ADP and anorganic phosphate. The I stands for inorganic. Why is that so important? Well, suppose, let's look at the first reaction of the glycolysis. Once glucose enters the cells, it enters the cell by a particular transporter. I guess you have looked at it, how this is taken up, glucose. And this transporter basically can allow glucose to enter the cell, but can also allow glucose to exit the cell. So the importance of the first step of glycolysis is that you modify the glucose molecule in such a way that it cannot leave the cell anymore. And that is by attaching a phosphate group to glucose. So you need an anorganic phosphate, the glucose molecule, and you generate glucose 6-phosphate. Now, as you all know, all chemical reactions are dealing with energy. Either you have to put energy into it, or you extract energy from a reaction. And that amount of energy that is involved in such a reaction is described by the change in the Gibbs energy, delta G. And for this reaction it says it's plus 3.3 kilocalorie, meaning that in fact you have to put energy into this reaction, 3.3 kilocalorie, to drive it into this direction. Now, I'm not going to bother you with a lot of thermodynamics, but the thermodynamics says that once a delta G, and the delta G is determined simply by the kind of molecules we are looking at, and also at their concentration at this particular moment. Now, once the delta G is smaller than 0, is negative, then it means that the reaction proceeds spontaneous. So in this direction, from left to right, it doesn't go. This reaction goes spontaneous from right to left. So in order to imagine, if I would have a bottle of glucose 6-phosphate here, and I would wait long enough, then finally it will go into glucose and 3-phosphate, because that's where the equilibrium is, on that side. And that's not what we want, because we want to produce glucose 6-phosphate. So how to solve that problem? We do that by coupling this reaction to another reaction, and that is the hydrolysis of ATP into ADP and inorganic phosphate. And that's a reaction which liberates 7.3 kilocalorie. So this reaction proceeds spontaneous from left to right. Now, by coupling these two reactions, and the coupling is, among other things, done by the presence of an enzyme, by coupling these reactions, you can actually add up those two into a new reaction, which is glucose plus ATP plus water gives glucose 6-phosphate plus ADP, and then also the changes in energy you can add up, and then you see I have a delta G of -4 kilocalorie. So this reaction proceeds spontaneous from left to right. However, there's one problem. It takes quite some time to establish the equilibrium, and therefore that's also the important thing of an enzyme. The enzyme that's doing this, I think it's, yeah, that's either hexokinase or glucokinase, takes care that this equilibrium is set at a reasonable time. Yes, is it clear to you? So, and once the concentrations are such that the delta G becomes 0, because you can imagine it's now -4 kilocalorie, but as I said, it depends on the concentrations. Once the concentrations are such that the delta G becomes 0, then the reaction goes as fast and as much from left to right as from right to left. So then we are in equilibrium. Okay, now sometimes you need to convert glucose 6-phosphate back into glucose and phosphate. We are not using, or the system is not using this reaction then, but a different reaction with a different enzyme. Yeah? So we are not generating ATP here. We are simply splitting off phosphate from glucose 6-phosphate. Completely different reaction. There is a difference between hexokinase and glucokinase, but I will come back to that a little bit later. Now, do you know what turnover means? Turnover of a molecule. That means that you can imagine that during the day you hydrolyze ATP, but you also produce ATP. And the number of molecules, or the amount of ATP that you hydrolyze, but also resynthesize during a day, that's what we call turnover. Well, not necessarily during a day, but the number of molecules that you hydrolyze and that you resynthesize, that's the turnover. If you would look at the amount of ATP that is going on into our body during a day at rest, what do you think? Would that be expressed in milligrams, in grams, or in kilograms? Just guess. What do you think? Do you have an idea on that? Would it be grams, kilograms, tons? Well, since you're not going to give me the answer, I will give it. It's 40 kilo a day. That's quite a lot, isn't it? So don't think that you lose 40 kilograms during a day, because you're also building up 40 kilograms a day. It's a stable situation. It's what we call turnover. And even during excessive exercise, like a sprint, it's a half a kilo per minute. So you can imagine the amount of, if you count that on number of molecules, it's an enormous big number. Okay, that's one important thing which we call coupled reactions in metabolism. Now, another thing that you need to know is about redox reactions. And I know everyone hates redox reactions. Secondary school, it has to do with oxidation and reduction and with electrons. Just to have an idea on what you know already about this. Oxidation. You may choose. Is that giving away electrons or taking up electrons? Oxidation is taking up electrons. So a compound is oxidized once it takes up electrons. Who's in favor? Yeah? 1, 2, 3, 4. About half. So the others agree that it is taking away, giving away electrons. Or don't have an opinion. Well, the first, although you nicely said, okay, it's taking up electrons, unfortunately, it's the other way around. This is what you really have to keep in mind. Oxidation, when a compound is oxidized, electrons go out of that compound. And a compound is reduced once it's taken up electrons. Yeah? Now, electrons you never have seen unless you're going maybe to look into extreme physics, but you never have seen a bottle with electrons, right? Electrons you have to capture or put away somehow. And electrons in metabolism are, oops, that's not what I wanted to do. Electrons in metabolism are taken up by a compound which is called NAD+, nicotinamide adenine dinucleotide. Usually the electrons are taken up from a compound together with 2 protons, so it's like extracting a hydrogen molecule from that compound. But in any case, it's an oxidation reaction because you extract electrons, and the electrons are put into NAD+, and that becomes then NADH and H+. And usually in metabolic schemes, these things are left out, and you only will see NAD and NADH. Yeah, they even sometimes leave out the plus, the charge on the molecule. But that's for you to remember, it always is dealing with electrons. Now, that means that NAD+ is an electron acceptor, and NADH, of course, is then an electron donor. That will be clear. We have another compound, which is called FAD, flavine adenine dinucleotide. And FAD can take up electrons together with 2 protons and then is turned into FADH2. There's yet another component which looks very much like the NAD+, and it's called NADP+, but you see already that I switched the direction of this reaction, because NADPH differs from NADH simply by 1 phosphate group, and that phosphate group tells the molecule, so to speak, that NADH will go to bring the electrons to the respiratory chain or the oxidative phosphorylation in order to produce ATP. We will see that later. But NADPH is not meant to go into the respiratory chain. NADPH is actually a store for electrons in order to allow reduction reactions to occur. Once you have to build up molecules and you need electrons to reduce compounds, to build them up to bigger structures, you sometimes need electrons. And those you extract from NADPH. Like, as we will see later on, in the fatty acid synthesis, you need a lot of NADPH to provide electrons. So again, here we have the electron donor, which is NADPH, and the electron acceptor, which is NADP+. Is this clear? So this is also
Which direction is oxidation and which is reduction?
well, yeah, that's a good question. You ask which direction is oxidation and which direction, what particular reaction you mean? For the last one or
Just
any reaction? Okay, now I have explained, you should be able to answer the question. So if I go from left to right, is that oxidation or reduction? For the NAD+ molecule, right? It's reduction, huh? NAD+ takes up electrons. So there's another component which is not shown here, yeah, but there may be another component here which donates the electrons in order to let it be taken up by NAD+. And that component that donates the electrons is oxidized. Yeah? So if a component is oxidized, there's always, at the same time, another component that is reduced. That's why we call it redox reactions. They always go together. Yeah? Because the electrons have to be generated somehow. Clear or not? Maybe you have to let it settle down a little bit. Yeah? It's more maybe language than really knowledge. You can oxidize and you can be oxidized. That's different things with electrons. Okay? Now, this is a slide very brief. I will not go into this very much, but to focus you on the fact that in whole metabolism we also are dealing and essentially require vitamins. And I give you one example of the first vitamin that actually was discovered, the vitamin B1, discovered by a Dutchman who actually received the Nobel Prize for his work. But he didn't do all the work. He initiated the work. He had the principal idea and the concept behind this all. There was this person who it had to do with the rice. You know, people eat white rice for a very long time, have the risk of developing beriberi, which is a neurological disorder with weakness of the muscles. And it was found out that in the bran around the rice, that part contained a compound which finally appeared to be vitamin B1. But this Polish biochemist tried and extracted that particular compound from the bran and came out with a compound which he called that it was a vital amine. So that and vital amine then finally turned into vitamin. Yeah? So that term was coined by this person. But what he did not know was he thought he had found that particular vitamin that this person was looking for, but it appeared that he finally actually isolated vitamin B3 and not B1. And the B1 was later on discovered and structure was resolved by this person, Robert Williams, a biochemist from the United States, and then it appeared to be this compound. But okay, this was just to stress you the importance of vitamins, because as you know, there are much more vitamins. But just to go back to the previous slide, niacin is a precursor of nicotinamine adenine dinucleotide, which is vitamin B3, and the flavin in the flavin adenine dinucleotide is coming from vitamin B2. So you can imagine if you're lacking these compounds, that the whole metabolism becomes under stress. It doesn't run, basically. Okay. Finally, about the general concept is about regulation of metabolism. Well, that can be done simply by changing the amount of enzyme by synthesis and degradation. That's not difficult to imagine, I guess. By controlling the activity of the enzyme, and that can be done by allostery, where some kind of products, a product here is derived from a substrate, but there can be another interaction with other molecules on that same enzyme, and that enzyme activity can then be influenced, modified. So I'm not going much into detail about this, but it's just to tell you that it's there. There's also hormonal control. So if you have a hormone which binds to its receptor, and you get a signal intracellular, and very often we are dealing then with phosphorylation reactions. So proteins, enzymes that become phosphorylated become more active, or sometimes when they are phosphorylated become less active. That's also possible. So that's also a way to modify the activity and manipulate the metabolism in the cell. And then there's also with the energy state of the cell. That's the amount of ATP that is present in the cell, and ATP, and it's a variant of the allosterase, that once ATP binds to the enzyme, it also modifies the structure of the enzyme and modifies the activity. So that's another thing. And then of course there's one final thing about regulation, and that is that things are organized in the cell. It's not just one soup of all enzymes and compounds, but as you know, we have a lot of intracellular structures, and each of these intracellular structures has its own part in the whole metabolic pathway. Like for instance the mitochondria, as we will see later, have very specific parts of the whole metabolism dealing with. Okay, now we are going to look at the degradation pathways, and since I asked you the question earlier, which pathway do you usually start when you look at metabolism, that's the glycolysis. Now first some nomenclature, but you can read that back from the PDF that will be available for you later on. Glycolysis, that's from glucose to pyruvate. Glycogenolysis, lysis always means breaking down things. So then you have glycogen, that's degraded, glycogenolysis. Genysis means that you're making something. So from glucose to glycogen is called glycogenesis. And similar, from pyruvate to glucose is gluconeogenesis. So you need to know some of these names. Well, not some, these are the general names for the carbohydrate metabolism, and I think you need to know them all. Now, the first thing I want to show you is the glycolysis, which was mostly investigated by these two people, Meyerhof and Emden, and that's why it's also called the Emden-Meyerhof pathway, but that's not less known, I guess. First thing that you want to do is, as we already discussed, you want to change glucose in such a way that it cannot escape from the cell anymore. So we attach a phosphate group. Now, you see glucose 6-phosphate. I don't know how familiar you are with the organic chemistry from secondary school, but you know we have this is the glucose molecule, that's the oxygen, and this is the first carbon atom, the second, the third, the fourth, fifth, and there is carbon atom number 6. So glucose 6-phosphate means that we attach a phosphate group to the 6th carbon atom. Then via fructose 6-phosphate we go to fructose 1,6-bisphosphate. As it says, we need another phosphate group to be attached, this time on the first carbon atom. The meaning of this is that we now can split up this molecule in two equal parts, which are called glyceraldehyde 3-phosphate. So you have to remember or keep in mind that for all the reactions that go behind this line, so from one molecule of glucose we have to multiply everything by 2. Yeah, if we want to do some counting on number of ATP molecules that we generate, from this line on we have to multiply by 2. Now you can imagine or appreciate that the phosphate needs to come from ATP, so this is the first ATP that we have to put in or to use for the glycolysis, and here is the second one. It's a bit strange, isn't it? Because we want to extract energy from glucose, and what we do, we start with putting in energy. Well, you can consider that as the starting engine. Yeah. Once it runs, you can produce the bulk of energy. And there we go. I have simplified the scheme, so not all intermediates are presented here. That's not necessary for you to know, but you need to know a couple of important steps. For instance, the glyceraldehyde 3-phosphate is an important one because that's like a cross-section. From there on you can go into different directions, as we will see later. So some of these molecules are really important to remember. So in some of these steps, in one of these steps, we liberate energy, and the energy is put into a molecule of ATP. Then here in the final step, going from phosphoenol pyruvate to pyruvate, we also generate energy into ATP. As you see, I also indicated not the whole structural formula of all these components, but what helps a lot in having a good view on metabolism is just counting carbon atoms. So glyceraldehyde is a molecule with 3 carbon atoms. So you know it's derived from that you derive 2 molecules of glyceraldehyde 3-phosphate from 1 molecule of glucose. There's another thing, and that is we have an oxidation reaction here. The first step, we oxidize, and the electrons that you extract now from this molecule have to be taken up somehow, and the somehow here is into NAD+, which then becomes NADH. Clear? So this again is a reduction reaction from NAD+ to NADH, but this is the oxidation reaction, and there we have the redox reaction, the redox couple. Yes? Now, once we have generated pyruvate, and actually I should first stress that this, what you see here, is the glycolysis, no more, no less. Yeah? So after the glycolysis, we go on, and the pyruvate is then taken up by the mitochondrion, and now I can point you at the different colors in the rest of my lecture. This color, the bluish color, is always the cytoplasm, the cytosol, and this yellow-orange color is always the inside of the mitochondrion. So that's the compartmentalization I will be dealing with in my presentation. So we have to enter the mitochondrion. Then in the mitochondrion, the pyruvate, which consists from 3 carbon atoms, will be degraded into an acetyl group. An acetyl, as you may know, I hope, is coming from acetic acid. Acetic acid, something which you have maybe in the cupboard of your kitchen, to put on the salad for the dressing, etc. We have lots of acetic acid in our body, but not as free acetic acid, but attached to what we call coenzyme A. So that's a kind of a basket to put in acetic acid units. Now, since acetic acid is composed or consists of 2 carbon atoms, we know that this reaction somehow has to get rid of 1 carbon atom. And to get rid of 1 carbon atom you can do by extracting carbon dioxide from the molecule. So the enzyme that is doing this is called pyruvate decarboxylase. This is to stress you that all these names of these enzymes, they look very complicated on first sight, but if you really look at the name of the enzyme, it's usually a very logical name. It says the substrate, pyruvate, and it says what the enzyme is doing. Decarboxylase, extracting a carbon atom from it. Yeah. We can also call it pyruvate dehydrogenase. You extract a hydrogen molecule, because this is also an oxidation reaction. So you take away 2 electrons and 2 protons, an H2 molecule, so to say. And that goes into NADH, same as what we have seen here. Yeah? Is this clear? Then acetyl-CoA, as we will see soon, will go into the citric acid cycle. And in the citric acid cycle we have a stepwise oxidation, we will see it in a minute, where we generate again a lot of electrons that are gathered into NADH. Then all these electrons collected into NADH will brought together in the respiratory chain or in the oxidative phosphorylation, and that's the site where the big amount of ATP is produced, as we will see. So this is the whole scheme of oxidizing glucose in order to liberate energy. There we have the vitamin B1, that is an essential part of the decarboxylation reaction. Now, I think I skipped this because for the sake of time it would take a little bit too much to explain the difference between hexokinase and glucokinase in the liver. Just have a look at it. If you don't understand it, please ask me next time, next week, that we can go into this. Yeah, it's an important concept though, but time is lacking now. Now, what would happen if we don't have oxygen? Well, something which Louis Pasteur already discovered when he studied yeast, because yeast you can grow under aerobic and anaerobic conditions, and he concluded from his experiments that if you grow yeast under anaerobic conditions, the yeast uses up much more glucose. And why would that be? Now, I'm going to explain you that, or it will be clear to you once this has been explained. What will be the problem if I don't have oxygen here? Then, of course, this whole business cannot run. That will be clear, I guess. Yeah, you have nothing to let the oxidative phosphorylation run, and then that means that also the citric acid cycle is blocked, and it also means that this reaction is blocked. So you end up with pyruvate. But okay, we didn't do the final calculation yet, but how much ATP do I generate from the glycolysis in total? So how much ATP is here generated? 0? 2? Why 2? Exactly, yes, very good. So here was this line. Yeah, I left it out here just to check whether you are still present. Here you have 2 molecules of ATP that you have to put into glycolysis. Here are 2 molecules of ATP coming out of glycolysis, but you remember this was multiplied by 2. So I finally generate 4 molecules of ATP, subtract 2 molecules, left net 2 molecules of ATP. So there's nothing wrong. I can simply, and that was what Pasteur saw, if I now lack this amount of ATP, then I let this reaction run at a higher speed in order to generate sufficient ATP. And that's in fact what happened in the yeast. But what is the problem? What would be the problem? Because it doesn't go by itself. What problem do I run into? Who is clever enough to explain this? There will be a problem. What do you know about anaerobic glycolysis? What are you producing? Lactate. Why are you producing lactate? What's the use of that? Because the lactate will be generated from pyruvate, as we will see, but it doesn't generate ATP. So why is the organism, the cell, going over to produce lactate under these conditions? No clue? No, the lactate is not used any further. Once it's used, it's simply going back to pyruvate and then either going up or going into that direction once there is oxygen present. No, you run into a problem here. What would happen, do you think? Sorry, a bit louder. There will not be any more NADH, but I'm producing NADH. Maybe you mean something else, because you're close. Yeah, you have a certain amount of NAD plus in the cytoplasm, but once all NAD plus is converted into NADH, and there's nothing to regenerate that because this doesn't run, then it stops. Now, how can we regenerate NADH from to NAD plus? What kind of a reaction do I need for that? I need to go from NADH to NAD plus. Is that a reduction or an oxidation? That's an oxidation reaction. Very good. So electrons are liberated. Yeah? Where do the electrons go? It's not a difficult question, just continue thinking. Where would the electrons go? That I take out electrons from NADH, which then becomes NAD+, fine, because then I can run this reaction again. But where do the electrons go? They go into pyruvate. What kind of a reaction would that be then? Electrons off. That's a reduction reaction. And what are you generating then? You're generating lactate. So lactate, in fact, is reduced pyruvate. Yes? And that's the only reason why a cell produces lactate. No more, no less. Yes? Well, I guess this is clear. Then I'll continue. This in fact was found out, because it's easy said now once you know everything, but it was quite difficult to find out this whole business, by Hill and Meyerhof. Now we enter the citric acid cycle, and again you see a picture of someone, and this is Mr. Krebs. who got the Nobel Prize for the work on the citric acid cycle. What's happening in the citric acid cycle? You remember we were left with acetyl-CoA. From the pyruvate, decarboxylation, we have the acetyl group, and the acetyl-CoA, then the acetyl group, the CoA is just something, an arm to put it through. But the acetyl group is going together with oxaloacetate, which is a component consisting of 4 carbon atoms. Adding up the 2 carbon atoms from the acetyl group, you end up with citrate. The citrate, C6, is then converted into isocitrate. That's nothing, not much important. Well, it needs to be done, of course, but no energy is involved in that. And then the isocitrate is converted in the alpha-ketoglutarate. And this is a compound which consists of 5 carbon atoms. So what kind of reaction would we have here? Like we had with the pyruvate to acetyl, a decarboxylation. Yeah, we have to extract a carbon atom from it. And then I'll continue. If you go from alpha-ketoglutarate to succinyl-CoA, and the CoA again is the same as what we have here. That's just a basket to put in the succinyl group. You go from C5 to C4. So again, a decarboxylation reaction. Now these decarboxylation reactions liberate carbon dioxide. So you can see already where the carbon dioxide is appearing once you combust glucose. Yeah, if you combust glucose, you get finally carbon dioxide and water. All the carbon dioxide is generated here and in the step here before from pyruvate to acetyl group. We still have to figure out where the water is coming from. Now, these were decarboxylation reactions, but also simultaneously oxidation reactions. So the electrons extracted from these reactions are converted or brought into NADH. Same is here from malate to oxaloacetate. So you see the number of carbon atoms doesn't change, but you again, it's an oxidation reaction where the electrons are put into NADH. And here is one reaction where we have electrons extracted, but now the electrons are not put into NAD+, but into FAD. Because to put electrons into NADH or FAD+, in NAD+ or FAD, also requires energy. And the amount of energy that is liberated here, here, and there is enough to put the electrons in NAD+, but the amount that's liberated here, that amount of energy is not sufficient to put it into NAD+, but sufficient to put the electrons into an FAD. Is that clear? Because you will see that back in the oxidative phosphorylation, that from this component you can generate a little bit less energy than from NADH, as we will see. Now this is the whole citric acid or grape cycle, except for one thing, and that is that we have also here energy liberated, and the energy that is liberated here is not converted into ATP but into GTP, but the GTP pool in a cell or in the matrix of the mitochondrion is in equilibrium with the ATP pool. So you can, well, basically also consider this as ATP. Now we enter the mitochondrion. Now you know about the structure of the mitochondrion, I guess, that it consists of 2 membranes, an outer membrane and an inner membrane with a lot of cristae. And in those cristae we have the enzymes that are required for the oxidative phosphorylation. Now what are these enzymes doing? Again, here we get someone who got the Nobel Prize for this, Peter Mitchell, for his work on finding out how the mitochondrion is dealing with the NADH to liberate ATP or to synthesize ATP. In the inner membrane of the mitochondrion we have a number of enzymes in complex brought together. These are the cytochromes, and the cytochromes have the characteristics that they can go from an oxidized to reduced state and back. So they make a kind of a circle. So they give away electrons, they take up electrons, they take up electrons, give away electrons, and cycle-wise. And here we come in with NADH. The NADH now liberates 2 electrons and then is converted into NAD+, which then can be used again, so it's regenerated, that's fine. That's what we wanted. The electrons are now taken up by the first cytochrome, the NADH-Q reductase, and that's going from its oxidized state into the reductive state. So it has taken up electrons here. Then it goes from the reduced form back into the oxidized form, and the electrons are now donated to the next cytochrome, the QH2, and then same thing happens, and the electrons are donated to the cytochrome C1, and then it's going to the cytochrome C, and it's finally going to the cytochrome oxidase. And then, what then? Because where does the cytochrome oxidase leave the electrons? Because if this one doesn't give away the electrons, then everything stops again. Now, for that we have oxygen, and for the stoichiometry, to have the proper number of atoms, I have used half a molecule of O2, and that takes up 2 electrons and becomes an O2 minus molecule. And the O2 minus is just a part of water. Yeah, we have protons here around here. So 2 protons and oxygen 2 minus will be water. So if you realize what I'm saying now is that water in fact is oxygen that has taken up electrons. Yeah? It's reduced oxygen, in fact. Okay. Why do we do this? Well, we don't do it. Why is the cell doing this? At some points of these reactions, the reaction is coupled to the transport of protons from the matrix of the mitochondrion to this inner space in between the 2 membranes of the mitochondria. And that happens also here and here. And so you can imagine that by doing this, by letting the electrons flowing down here along the cytochromes and to oxygen, you generate a proton gradient. And a gradient always contains energy. It's like bringing up a bucket of water on top of the hill, and then you can let the water go down, and then the water that goes down can charge or drive a mill that produces energy. So you have brought up the bucket, potential energy is increased, and finally you convert that into mechanical electrical energy. So the protons now, this gradient can be used by allowing the protons to go back to the matrix, that's what they want, via a protein which is called ATP synthase. And the energy that's liberated during that process can be used to attach an inorganic phosphate to ADP in order to produce ATP. That's how the cell produces the bulk of ATP. Is this clear? Now, as I said, we also have this molecule of FADH2, and I already said that it contains a little bit less energy, and that's what you can see here, because this FADH2 cannot give the electrons to the first cytochrome, but only to the second one. So you're missing a little bit of that proton gradient, and that's the reason why from FADH2 you liberate a little bit less ATP than from NADH. It's a detail, but just for you to know. Now, there's one more thing, and that is that, of course, we need to bring this ATP, we need, of course, a lot of ATP in the mitochondrial matrix, but we also want to have ATP in the cytoplasm for all sorts of reactions. So we need to bring that ATP along via these membranes to the cytoplasmic space, and that also requires a little bit of energy. It requires a little bit of that proton gradient that we produced here. So finally that gives the number of ATP molecules that you can produce per molecule of NADH. Now in the old biochemistry books you will see that that number is 3 molecules of ATP per molecule of NADH. In the modern biochemistry books they have taken into consideration this part, and then these books say that 1 NADH gives 2.5 molecules of ATP. Yeah, that's for you to know once you find that in the books. And that means that from glucose, what you probably learned in secondary school, if you digest or metabolize glucose, you liberate either 36 or 38 molecules of ATP. Nowadays, because of this correction, it's 30 or 32 molecules of ATP. Yeah, and that the basis of that is depicted here. Now, this is a kind of a cartoon, not just to let you relax a little bit, but well, what would you think these things would have to do with each other? We see here a movie star from the previous century. It's Rita Hayworth for the, well, you probably don't know, I didn't know either. This is of course the symbol for, what is it? Symbol for? Are you awake? Yeah, you are awake. Explosives, right? And this is a brown bear. What have these three elements to do with each other? Well, I'll tell you. We start with the explosives. Halfway last century, there was a lot of industries going on in producing explosives. And the employees in these industries were not feeling well. Losing weight, feeling tired, malaise. You know that word? Feeling awful. And this was one of the first situations in which what we call in the Netherlands the arbodienst came into the picture, that an organization was going to look into what happened to these employees. Why do they get sick? And they found out that they were working with a compound which is called 2,4-dinitrophenol, DNP. And they found out that this compound is a very lipophilic compound. You know the word lipophilic? Yeah. So the compound goes into fat tissue, but goes into all fat materials, also in the membranes and in the membrane of the, also in the inner membrane of the mitochondria. And that compound had the characteristic that it allows protons to move through the membrane back to the matrix. So what happened was that the gradient that you build up here in order to generate ATP, that gradient dissipated via the 2,4-dinitrophenol. And so it didn't produce ATP. And that's why these people became so sick and did lose weight. Because why did they lose weight? They wanted to generate ATP. So the whole metabolism was set in a situation that mobilized as much as possible energy from all tissues that we have, including the fat tissue. So you're burning down your fat tissue, but with no use because it didn't produce ATP. Okay, once finding that out, the circumstances for these employees were improved, so they were no longer sick, that's fine. But there was a smart guy and thought, maybe we can use this compound as a slimming agent. People who have grown too fat, obesity was not as big a problem as it is now, but it was already a problem also in the previous century. If you have too much fat tissue or you're too heavy, maybe 2,4-dinitrophenol will do the business and will get you rid of a lot of fat. So there was this lady, Rita Hayworth, who was a little bit more fat, I think, than in the previous slide, and she started to swallow this medicine, slimming agent. But you can imagine she had too much fat, so all that material primarily first went into the fat tissue, her fat tissue. So it was useless. It didn't work. But she kept on taking this medicine, and then finally the medicine reached the concentration, saturated in the fat tissue, then it went into the mitochondrial membrane. And so it switched on, in fact, this situation. And you can imagine what happened. Then the fat will be broken down, and all the 2,4-dinitrophenol that was present in rest in the fat tissue was liberated and went into more and more mitochondria. So what would you think what happened to this lady? Very fast metabolism. She didn't die, but almost. Yes, okay, but what was the first thing that happened? All these reactions going on. What is always happening with most reactions, that the energy that's liberated, there's also a little bit of energy liberated in the form of heat. So her body temperature went up to above 40 degrees. And as you know, that's a dangerous situation. Now, she survived the whole exercise, by staying in a bathtub for 3 days, the bathtub filled with ice cubes. Yeah, that's the story. It's an anecdote, but okay. Now, what has the brown bear to do with this whole story? That's a question for you. And I could have said other, I could have shown other animals as well. Yeah, yeah. A brown bear, as a lot of other animals are doing in the winter time, they go asleep. Yeah, in a quiet place, but usually a cold place. Yeah, not so cold, but pretty cold. And so they need to keep their body warm, and that's done with brown fat tissue. Yeah, you all have heard of brown fat tissue, I guess. And what's the essential of brown fat tissue? Brown fat tissue contains mitochondria, which do not have 2,4-dinitrophenol, but watch, they have the so-called uncoupling protein. So that's a physiological, natural situation. A part of part of our fat tissue is called brown fat, and the brown fat has mitochondria which have this protein. Now, we don't have that much brown fat, we as adults. Babies do have brown fat. You know where it's situated mostly? Or lots of? We still have brown fat. Where do we have our brown fat? It's not very much known, huh? Well, I didn't know it either a couple of years ago, but it's between the, it's here. And I can show you this on the next slide. This is a PET scan. It's a particular technique where you can visualize metabolic activity. And here you have the situation when the environment is warm enough, the temperature is high enough. So let's say summertime for the bear, nothing's visible, but in wintertime you see these activities around the shoulders, being high metabolic activity. This is because of the brown fat tissue. Okay, I'll move on. To frustrate you a little bit, we have now had this part of the whole metabolic scheme. So we quickly move on. Yeah. So this is the glycolysis, citric acid cycle, and respiratory chain. This is basically what you have been dealing with, I think, in secondary school. But we need to do a little bit more. We go to have a look at fat, and degradation of fat, and synthesis of fat later on. Now, again, we have a number of names. If we go from fat to fatty acids, a process we call lipolysis. This is fatty acid oxidation from fatty acids to acetyl-CoA. That's fatty acid synthesis, no specific name for it. And that's lipogenesis. Yeah, the production of lipids. Now, fatty acids are coming from fat, and fat, most of the fat that we know is fat that is in the form of triglycerides. So we have a glycerol backbone, and the glycerol is 3 molecules, 3 carbon atoms with an OH group, and each OH group has a fatty acid attached to it, and a fatty acid is usually a very long tail of carbon and hydrogen atoms, a very apolar molecule. Now, that molecule is hydrolyzed by lipases into glycerol, and the glycerol, as we will see, easily can enter the glycolysis or the gluconeogenesis. So it comes in, let's say, halfway the glycolysis and can go down to acetyl group, or it comes in halfway the gluconeogenesis, which looks very much like the glycolysis, but the other way around. We will see that later. But we will focus now on the fatty acids. Now, this is the form of a fatty acid, so you have a very long tail of carbon and hydrogen atoms, saturated or unsaturated, meaning that you have single or double bonds here. You all have heard of that. And you have a carboxyl group here. Now, what's happening to that molecule? That molecule will be degraded stepwise in the mitochondrial matrix by splitting off C2 units. Each cycle, a C2 unit is split off and will appear in the form of an acetyl group attached to the coenzyme A. Yeah, so you start with the fatty acid, attaching it to a molecule of coenzyme A, and then go into the cycle and then stepwise produce these acetyl groups, and then of course after one cycle you have the new molecule with one acetyl group less. This is called the beta oxidation because it's on the beta carbon atom that the molecule is split. It's also called the Knoop cycle because Franz Knoop was a German investigator. He did very elegant experiments to elucidate this whole cycle. Did you have a question?
Can you have, the start of the citric acid cycle if you didn't have beta oxidation to get acetylcholine? Or does that
Speak a bit louder, I can't hear.
Can you have the start of the citric acid cycle where you have the addition of acetylcholine if you didn't have beta oxidation?
Well, I don't know whether I understand the question quite well, but the acetyl groups that are generated here will indeed go into, or can go into, the citric acid cycle, if that is your question. Yeah. Yeah.
So if that cycle, that process is inhibited for whatever reason,
Exactly.
citric acid
Exactly. That's a very good remark. So if you inhibit the citric acid cycle, or if there is no citric acid cycle, some cells do not have a citric acid cycle. Which cells? Okay, we come back to that later. We leave that for as a surprise. Yes, exactly. Red blood cells. They don't have mitochondria, so this already preludes on what I'm going to say later on. But if you don't have a citric acid cycle, or if you don't have oxygen, this process will not run. It might run once, but then you have taken up all the NAD plus and everything that's in the cell compartment, and then it stops. Okay. This is a stepwise process, as I said. Also stepwise oxidation, and the electrons that are coming from the oxidation reaction are put into FAD or NADH, NAD plus. There's FADH2 or NADH. Yeah. Now, of course, as we already said, the acetyl group can go into the citric acid cycle, where more NADH is liberated, and then all the NADH and FADH2 that is collected by doing this stepwise process will go into the oxidative phosphorylation in order to liberate ATP. Oh, okay, yeah, that was a bit quick, but we were left with one problem, because we have this fatty acid here in the cytosol, and that needs to go to the mitochondrion. Now, there's a particular reaction for that, which is depicted here. You don't have to know the details of that reaction, but I show it here because it's going via a carnitine ester, and there are some diseases in which the enzyme that is doing this is lacking, and then you can imagine that these patients do have a problem with the fatty acid oxidation, and do have a serious problem, because fatty acid oxidation is important for our energy production. So now we have seen this part of the scheme. It goes a little bit faster now. So you can imagine the fatty acids going down here and ending up in acetyl groups, going into the citric acid cycle, producing ATP, and the glycerol can go the same direction. Well, the same, not the same, but glycerol can go via the glycolysis in the same way to produce ATP. Now a little bit of calculation. If you look at the glycolysis, you can simply look at how much energy is produced at each step, and for the glycolysis it means that you finally end up, if you calculate all the NADH and etc., and what you produce per NADH and FADH2, then you end up with 32 molecules of ATP per molecule of glucose. If you take, for instance, a fatty acid like palmitic acid, which consists of 16 carbon atoms, so you can do 7 times the clip in the cycle, clipping off 2 carbon atoms, producing all the acetyl groups, then you can see that there is a little bit of ATP that you have to put into the fatty acid. I forgot to tell you that last slide, but similar to the glycolysis, you first have to put in a little bit of ATP to allow the fatty acid cycle to go. So that's the same here. The beta oxidation, 7 cycles, 7 NADH, which liberates 17.5 ATP and 10.5 ATP from FDH2. And all these acetyl groups that are liberated finally give you 80 molecules of ATP. So I get 106 ATP. So this is much more than from glucose. But the comparison, of course, is not entirely fair because glucose only has 6 carbon atoms and here I have 16. So it would be more fair to calculate the number of ATP molecules per molecule of ATP, per carbon atom, excuse me, yeah? That means that for glucose, I have 5.3 ATP per carbon atom, whereas for fatty acids, I have 6.6 ATP per carbon atom. Huh? So that means that fat has much more ATP content than carbohydrate. Is that clear? Because that's the basis for the next slide. If you imagine in a human body, an adult human body, about 70 kilograms, we have about 100,000 kilocalories in store on triacylglycerides, in fat. We have about 25,000 kilocalories in proteins, but of course we don't use that as energy source, but it represents that amount of energy. Then for glycogen, even if all our stores of glycogen are complete, it's only 600 kilocalories, and free glucose only 40 kilocalories. Now if you realize that the amount of fat that we have in a body, in a just normal healthy body, so this amount of fat is about 11 kilograms. What would happen if I would need to store all these kilocalories, not in the form of fat, but in the form of glycogen? What would be the consequence of that? Now you have to imagine that fat is lipophilic. Lipophilic. It likes to be in a fat environment, so it doesn't want to have water around. Yeah? In contrast to carbohydrates. So glycogen, if you store glycogen, you also always store water as well. There's a lot of water attached to the glycogen molecules. Now that results in, if you would produce all triacylglyceride into glycogen to have the same amount of kilocalories, the body would be 55 kilograms heavier. That's not a good situation. Then you are simply tired by the end of the day by sitting and walking around. To stress this a little bit more, this little bird, do you know what kind of bird this is? No, it's not a swallow. It does have indeed a two-pointed tail. It's a stern, or tern I think in English. And what's the specialty of this bird? This is a champion among the birds. I think this... Yeah, it can fly for a very long time and it can fly very fast. I think the speed, the airspeed of this bird is about 60 kilometers an hour, which is quite a lot for a bird that weighs about 100 grams, I think. And what this bird is doing, it's spending part of the year here and then flies to the South Pole, spending a little bit of time there, flies back to the North Pole. So this bird actually migrates about 35,000 kilometers per year at an average day distance of 240 kilometers. That's a lot. And you need to have fuel for that. Now you can imagine if the bird only takes fuel as glycogen, the bird would be too heavy even to fly. So it really requires fat and fat tissue in order to accomplish this big journey. Yeah? So this is, I think, the best example to show how important fat is as energy source. Okay. Respiratory quotient. You have heard about this in block 1, 2, I think. Yes? So you may remember that that is the volume of carbon dioxide divided by the volume of oxygen. And you know what, if you combust or metabolize mostly carbohydrate, what would be the respiratory quotient? Because that's why you use it for, huh? Respiratory quotient tells you something about the fuel that you are using. what would be the fuel, or what would be the ratio CO2 over O2 when your fuel is only carbohydrate? It's an extreme situation, never occurs, I think, but no, carbohydrate. So like glucose. Just one, huh? Why is that? Well, you know it's one, that's already one good thing, but it's very easy. If you look at the circular formula, this is glucose, C6H12O6. You remember this from secondary school. You have had this, I think, in the third level of secondary school. This fiddling around with all these coefficients to have the balance of the equal number of carbon atoms and oxygen atoms. And that's what it says here. I need 6 oxygen molecules and I produce 6 carbon dioxide molecules to metabolize a whole molecule of glucose. And so the ratio is 1. Yeah? If you do that for fat, you have this situation. This is an example of the palmitic acid that we saw in the previous slide. You have 16 carbon atoms and you require 23 molecules of oxygen, meaning that I have a ratio of 16 divided by 16 divided by 23, which is 0.7. So if you're measuring the respiratory quotient and it's close to 0.7, you can see that the body is mostly degrading and metabolizing lipids, fat. It's a little bit more complicated, of course, because we have also proteins that can be digested and that give intermediate values. That's about the RQ. Now the production of keto bodies. I have to speed up a little bit, I guess. Keto bodies, most people think they are bad things in metabolism, but they are essential components of the metabolism. Keto bodies, what the diabetics are producing in excess. And if you have too much keto bodies, keto acids, yeah, the plasma, the blood becomes too acidic and you get a lot of problems. But under normal physiological conditions, keto bodies are important as well. Because from acetyl-CoA you can produce acetyl-CoA, so basically 2 of these molecules brought together. Then you go to this very complex molecule which has a very difficult name, 3-hydroxy 3-methyl-gluteryl-CoA, HMG-CoA. And that is converted to acetoacetate, and acetoacetate can be used by different tissues as fuel, because it can go back from here to acetyl-CoA. But it can also be converted into beta-hydroxybutyric acid and acetone. Now the acetone is of no use, that's just diagnostically. If you smell acetone from a people, from the breath of someone, then you know something is wrong. Should not be detectable. As I said, the acetoacetate can be used as fuel, can be brought back to acetyl group and then go back into the Krebs cycle. And then one more thing to stress with this slide is that this compound is an intermediate in the synthesis of cholesterol, and cholesterol is, as you know, should know, is a very important component because it's the basis for the synthesis of steroid hormones, for the production of bile acid, which we need for the digestion. It's a basic component of membranes, so cell membranes without cholesterol don't function well. It's a part of the lipoproteins, but that's just the way how it's transported, and it can be stored in the form of cholesterol ester. So cholesterol is not a bad molecule, it's really a molecule that you need. And now I think we can have a small coffee break, but I would really urge you to be back here in 10 minutes, because we have until 10:30 and we still need to do a lot. Or shall I continue? I don't know, because I don't want everybody to fall asleep. Small break? 5 minutes? Is that enough? So be back here at 10:05. have discussed now this little part of the metabolic scheme, the production of keto bodies and the production of cholesterol. Now we are going to look at proteins, still digestion or metabolism, degradation. For proteins, we have the different names like proteolysis, going from proteins to amino acids, and from amino acids to keto acids, that's what you produce. That's a process that we call transamination, where urine is produced. And from keto acids to produce amino acids is amino acid synthesis, and there we have protein synthesis. Now, what is the degradation of amino acids? You need to know, of course, the structural formula, the general formula of an amino acid. But you have had that, I think, already in Block 1.1, or you should know it from secondary school. You have a carbon atom, you have a carboxyl group, you have an amino group, and you have a residual group that varies among the different amino acids. We have 20 of those in the natural occurring proteins in the body. And what is happening upon degradation of the amino acids is that you convert an amino acid into a keto acid. Now, a keto group is characterized by this, a C-double-O, double bond O. So this amino group, C amino group, is converted into a CO group. And that's what we call a process which is called transamination, because the amino group is transferred to another keto acid that then becomes an amino acid. Yeah? So you have a keto acid and an amino acid. Let's say this is the amino acid, this is the keto acid, and after the reaction this one is the amino acid, and that becomes the keto acid. Useless, you would say, but not entirely, as we will see. The transaminases, the most well known are the ALAT and the ASAT. You may have heard of it already because it's used diagnostically. Alanine aminotransferase and aspartate aminotransferase. It's what the word says. Alanine is an amino acid, aminotransferase. You transfer it to another molecule. That's the name of the enzyme. And then alpha-ketoglutarate acid is converted, and that's an intermediate of the citric acid cycle, is converted into glutamic acid. Now, in order to make this not a useless process, because then you want to regenerate glutamic acid into glutaric acid again, otherwise, if you would not do that, then the citric acid cycle comes into problems, because you use up an important intermediate of the citric acid cycle. So in order to regenerate alpha-ketoglutarate acid in a different way, we have that's fun. We have this enzyme, glutamate dehydrogenase, meaning you extract electrons. And the electrons are going into NAD+, so that's, well, somehow energy production. But on the other hand, the amino group now is not transferred to another keto acid, but it's really extracted from the molecule, NH4+. This is what we call an oxidative desamination. This is the oxidation part, and this is the desamination part. And then you regenerate alpha-ketoglutarate acid. But of course, ammonia is a very poisonous component, so you need to get rid of it. And that's what you do in the urea cycle, which I will not deal with here. That's too complicated, takes too much time for now. So I have simplified this by this cyclic process, urea cycle. Ammonia is converted into urea and can be extracted via the urine from the body. But of course, it's a process in which you want to get rid of particular products, and that is going at the expense of energy. It takes 4 molecules of ATP to do that. Clear? So that's the degradation of amino acids that you see here. The keto and the keto acids that are liberated. So I go back here one more time to tell you what kind of components these are. I take a very simple example. and that is if you have the amino acid alanine, once you convert alanine to its keto acid, the keto acid that belongs to that is pyruvate. So this is to give you an example that what is the fate of these keto acids that that are liberated upon transamination of amino acids. They end up in different parts of the metabolic pathways. So for alanine you end up at pyruvate. If you take aspartic acid, you end up with oxaloacetate, an intermediate of the citric acid cycle. So each amino acid has its own pathway. Now we are going to look at the synthesis. And the first pathway for synthesis is this one. It's let's say a detour from the glycolysis. Here we have the glycolysis, glucose to pyruvate. But now from glucose 6-phosphate, we go into this direction. We go to 6-phosphogluconate. That's why this pathway is called the phosphogluconate pathway. That's the first substrate that is entering in this detour. And the meaning of this is that this is again a dehydrogenase. So you extract electrons. But now this time the electrons are not used to finally produce ATP. Now we want to have these electrons collected, stored for other purposes. And so we store them now in the form of NADPH. Yeah? NADP plus is converted to NADPH. Then there's a number of pathways, and this is a very diverse number of reactions. I'm not going to deal with that. The only thing I want to say is that apart from the fact that at particular other points in this pathway there's more NADPH produced, but there's also production of other sugars with other number of carbon atoms. For instance, ribose, C5, is coming from this pathway. And as you know, ribose is an essential component of DNA and RNA. So in cells where you have a lot of DNA and RNA synthesis, like the enterocytes, where you have continuous proliferation of these cells, you need to make a lot of DNA and RNA. And so these cells are very rich in this pathway. For the NADPH, as I said, that is important for synthesis, for instance for fatty acid synthesis. It's also important for repair of oxidative damage. And to illustrate that here, you know these kind of vegetables? In the Netherlands we call it tuinbonen. Internationally it would be fava beans. There are people that become seriously ill once they eat these kind of products, these fava beans. And severely ill means that they sometimes have to be brought in at the first aid center in the hospital. And if you then collect some blood from these patients and run a hematocrit, you have done a hematocrit in the skills laboratory, so you know what it is. Hematocrit, this is the normal situation, so you spin down the blood and you have about half the volume consists of the red blood cells, and the remainder is the plasma volume. If you do that with patients that are brought in the emergency after having eaten these kind of products, then you will see that the level of the red blood cells around here, if you look carefully you can see it, and the plasma column is completely red, meaning, what does that mean? If the plasma column is red, because that's not normal, right? It should be well, colorless, a little bit yellowish or yellow. A bit louder, I can't hear you. Yes, exactly. So your red blood cells lyse. And why do they do that after eating these fava beans? Red blood cells, of course, they transport oxygen. So they are prone to oxidation. And so the lipid components of the membrane become oxidized. But they have to be reduced again, because if you have too much oxidized lipids, the red blood cell loses its flexibility. And then they break into the capillary system. So you get hemolysis. So in order to repair that, you need a lot of NADPH. The reactions that are doing the repair mechanism require NADPH. Now the patients, or the people that have this problem are, well, patients, because they lack this enzyme, glucose 6-phosphate dehydrogenase. and it's worldwide. I think it's one of the first discovered genetic disorders, and one of the worldwide most occurring metabolic disorders. There are all sorts of variants of this disease, but the serious ones have this problem here. Okay, so that is this part. Yeah? DNA, RNA synthesis, and production of fatty acids, for instance, and repair of oxidative damage. Now, glucose. We sometimes need to make glucose, and glucose cannot be made simply by going from pyruvate back to phosphoenol pyruvate and then all the way up to glucose. That's impossible. Some of the reactions are going into both directions, but on particular points you need different enzymes. Now, pyruvate can be made from lactate, so once a cell has made a lot of lactate because there was a condition with less oxygen, like when you do a sprint, yeah, heavy exercise, then that lactate can be easily converted to pyruvate and still can be used to produce glucose, but not according to this way, but the pyruvate will enter the mitochondrial matrix. Pyruvate, remember, was a C3 molecule, and we used to go to the acetyl group, yeah, with the decarboxylation, but we are not doing that right now. What we do now is we want to go from pyruvate to oxaloacetate, which has 4 carbon atoms. So now we don't have a decarboxylation reaction, but a carboxylation reaction. We have to put in a carbon atom into the molecule, and that requires energy. So we have now the enzyme pyruvate carboxylase, which turns pyruvate into oxaloacetate at the expense of ATP and 1 carbon from carbon dioxide. Oxaloacetate, as you may remember, is an intermediate of the citric acid cycle, meaning that, for instance, amino acids or other components from the metabolism that enter in intermediates of the citric acid cycle can also be used to produce oxaloacetate, and oxaloacetate is the precursor of the synthesis of glucose, because oxaloacetate can be, via different transporters, brought to the cytoplasm, and in the cytoplasm oxaloacetate can be converted into phosphoenol pyruvate. So we have circumvented this problem by going that way. Yeah? Then from pyruvate we can go to glyceraldehyde 3-phosphate and the whole way up to glucose. Now there is, as I said, this point of the amino acids that enter the cycle at different instances, different spots. I want to stress, but I will come back to that next week, that from fatty acids, if I would ask you the question, can you make glucose from fatty acids? Well, if you look at this scheme, what would you say? Just use it as a route map.
Yes.
Then you would probably say yes. I agree with that. You would say yes, but you can already hear from my voice that the answer is no. Yeah? You would go like this. Yeah? You go via the citric acid cycle, oxaloacetate, and then all the way up in the gluconeogenesis. But look what happens. The acetyl group is 2 carbon atoms. These 2 carbon atoms enter the cycle here via citrate, but here 1 carbon atom is liberated, and here the other carbon atom is liberated. So it's gone. Yeah? So you cannot make glucose from fatty acids. It's simply gone before you end up at oxaloacetate. Yes, is that clear? Because this is the basis of, for instance, how a diabetic patient can go into a diabetic coma by producing too much keto acids. What's the question? No? But otherwise, if you have question, as I said, next week we will come back to this one more time. Now, of course, this is not the whole story, because as I don't know whether you saw what happened, if I press the button here, you see there, we need an enzyme here, different from hexokinase, that converts glucose 6-phosphate into glucose, and that's the glucose 6-phosphatase. Yes? This whole process takes energy. From lactate, per molecule glucose, I require 6 molecules of ATP. Can you see that calculation? Can I maybe interrupt? Are you still with me, or are you doing something else? Because it really disturbs me. Okay, but I told you, just tell me if it's all clear to you, then I go at a higher speed. Okay? Okay. So from lactate, I need 1 molecule of ATP, another molecule, so I need 3 molecules of ATP, but remember there was this line here that says to produce 1 molecule of glucose I need to run this part twice. Yeah? Okay. That's the production of glucose. That's going this way. So you see the scheme becomes more and more complicated, but now it may not seem as complicated anymore, I think. Now the production of fatty acids. Fatty acid synthesis is basically a very simple route. It starts from acetyl groups, acetyl-CoA, and you have a first reaction which is a carboxylation reaction. Again, that requires ATP, and you end up with malonyl-CoA, and then you continue that series of reactions to build up a longer molecule consisting of carbon atoms, more and more carbon atoms. That's how you build up your fatty acid. Those reactions require a lot of electrons. These are reduction reactions, and the electrons are coming from NADPH, which, as we have seen, are coming from the pentose phosphate shunt. You know, the shunt with the fava beans, to remember. Now, there's a problem, but because acetyl groups, acetyl-CoA, usually is present in the mitochondrion, and I have to bring it up here. The acetyl-CoA, by the way, in the mitochondrion, of course, is coming from different routes. It can come from glucose, yeah, via pyruvate. Acetyl-CoA can go into the citric acid cycle, but it can also go that way. So this is a scheme which shows you how you can convert glucose, or carbohydrates in general, into fat. Yeah, that's going via this route. Now, even amino acids. Yeah, if you take up a lot of amino acids, more than you need to build up your proteins, then you can convert them, and you can, via different routes, at least part of those amino acids, you can convert into acetyl groups, which can be used for storing energy in the form of fat. Now, the problem to get the acetyl group into the cytoplasm, I have solved by this way. That's a series of reactions. That's why it's a big arrow. But I don't go into the details on that process. The only thing is that in several of these reactions, we even produce more NADPH. So that's the only. There should be another arrow here saying NADPH is coming from this transfer of acetyl groups from the mitochondrion to the cytosol. And that's this part here, the building up of fatty acids. Now let's see, I think I'm almost done. Amino acid synthesis, that's a bit of a problem because we have 20 amino acids and each of them does have more or less a different pathway. So most of the amino acids, you can simply say, can be produced, of course, by the reverse of the keto acids that you make up in different routes, and the keto acids are then transaminated into the corresponding amino acid. And that's basically, I think, what you should remember, or try to remember from this. Apart from the fact that, of course, we do have most amino acids we can make ourselves, but there are a number of amino acids that you really have to take in via the food. Those are called the essential amino acids. And that's dependent. For a baby has a different number of essential amino acids than adults. And to show you how that can go, like pyruvate, I already told you, via transamination goes into alanine. Oxaloacetate, via transamination, goes into aspartic acid. Alpha-ketoglutarate acid, transamination, glutamic acid. So here you see some examples of how amino acids can be produced. Now to make the story a bit complete, the last slide is about nucleotide degradation and synthesis. You may appreciate that I didn't discuss about this yet, but to make the whole metabolism complete, it should be there. I think what you need to remember from this is some of the nomenclature that we have. Different names for the different parts of the molecule: adenine, adenosine, adenylate, like base, the ribonucleoside, and the ribonucleotide. The importance of the degradation is shown here, and sometimes there can be abnormalities in here. So we have enzymes, nucleotidase and adenosine deaminase. We produce upon degradation urate, and some of the deviations are that there is a disease which is called SCID, which is a problem in this enzyme. This is a severe immunodeficiency disease. You know about the boy in the bubble? You've ever heard of it? These children that are born, they need to be kept in a kind of situation that they cannot come into contact with the natural environment because of all the attacks that we get from outside that we are dealing with our bodies and our immune system. But these children don't have, basically they don't have an immune system, or not one that operates. And then if you produce too much urate, which also can occur in other diseases, you have what we call gout, which is a very painful experience if you have it. The synthesis is de novo synthesis from activated ribose, amino acids, ATP, carbon dioxide, and you produce ribonucleotides and deoxyribonucleotides. Or it's what we call a salvage pathway. That's really turnover of when you break down DNA or RNA, you can use the building blocks again to make the proper ribonucleotides or deoxyribonucleotides. And I hope, actually, this is more or less it. This is bringing you back to the case this afternoon. Yeah, you have a question about the previous slide? Sorry?
What is gout?
Yeah, I know the English-speaking people know what gout is. In Dutch, you're Dutch, I guess. Jicht. Yeah, it's urate crystals that urate is soluble, but once it can be possible if the concentration is too high that it crystallizes in the microvasculature, mostly of the extremities, because of the temperature. So those people, for instance, have, when they lie in bed and they have cold feet, then it may crystallize into the feet, and that is very, very painful. And one of the things to, apart from medication, one of the things sometimes to resolve the problem is just putting your foot in hot water. Yeah? Then it resolves again. That's about gout. Oh, sorry. Yeah, it was the end. So it's my last slide. So what you have to do this afternoon, I already told you, discuss the different tissues in relation to what I have tried to refresh in your memory right now on metabolism. And I can imagine that still will be difficult, but struggle with it. Try to study this week on this subject. That's the meaning, I think, of this week. And then next week we will come back and we will address things like, okay, what happens with all these molecules that derive at these different tissues, and what happens if you stop eating, if you start starvation? How are the metabolic changes, pathway changed in the different organs? How is it regulated? That's the meaning of next week. Yeah, is this clear? Because it's 10:30, that's just the time I got till the end of this lecture. No more questions for the moment? Okay, thank you.